Welcome everybody to our first December meeting of the IMAG MSM Working Group on Multiscale Modeling and Viral Pandemics. I need to remind you that the meeting is being live streamed on YouTube and it's being recorded for prior viewing, or subsequent viewing. And I appreciate you coming today. We very much appreciate our speakers. As always, we have Reinhard, who's going to be speaking today, and myself as co-leads of the working group, Jim Sluka and Bruce Shapiro, also working with us. You can always contact us with your suggestions for improvements, ideas, complaints. We have our usual social media. We definitely would like to publicize more. Please help us out. We have a Twitter handle. If people can announce and retweet for us, that would be very helpful. We have our wonderful archive of talks, which deserve more viewing than they're getting, and we would appreciate your help getting the word out. Are there any announcements before we start the seminars? No? So today, before we get started on the seminars, I want to give us a schedule for upcoming meetings. We have 2 talks arranged for next week, one of which is going to be on software development, which should be a bit of a different kind of talk from our usual. We have the rescheduled talk by Russ Taylor on robotics in the ICU, which we had technical trouble with a few weeks ago. Jane Parker, also from the UK, is going to be talking about her work on loss of smell and taste associated with viral infection, which should be interesting. We have 2 weeks off for Christmas break and New Year's, and then we will start out with the important talk on experimental work on trying to quantify relationship between viral load and infectiousness, which is something that, of course, connects the in-house modeling with the epidemiological modeling. We have other talks coming up, but we, as always, are looking for your suggestions for speakers. Feel free to suggest yourself again, even if you've spoken before, postdocs in your group, graduate students, or people you just want to hear. I would ask people to remember to mute their mics and hold your questions until the ends of the talks. Today, because of the time difference, Claire will present. We will then have time for her questions. Reinhard will present at the end of the hour, and then we'll hold the discussion of Reinhardt's material until the discussion session between 4 and 4:30. So to begin with, we're very grateful to Clare Bryant for coming to talk to us about conservation of host cell death in response to bacterial infection. People know that I don't do elaborate introductions. I've taken enough of your time, and I will turn the screen and the floor over to Claire.
Thank you very much. Hopefully you can see my slides. Can you see them as normal view, or do you see them as presenter view?
For some reason, it is presenting half of your slide.
Oh, that's
so there
weird.
Seems to be a format there. Now that's perfect.
That's perfect. Great. Okay. Good. Right. So thank you very much for inviting me to talk. It is somewhat late in the evening for me here. I've had my dinner, so I should be fueled and ready to go. And I have to say, it's the first time I've ever been asked to give a talk because of an article that appeared in The Economist. So that's a first. Mind you, it's the first time I ever had an article appear in The Economist about my science. So life is very curious sometimes. So what I'm going to talk to you about is some of the work that we did relatively recently, which I've been working on comparative biology for a long time, and I'm pretty sure it'll be the same in the States as it is in the UK, which is I'm a veterinarian and I'm very interested in comparative biology and the lessons that I can learn, but predominantly funding comes through work on mice and humans. So I've been fascinated for a long time about zoonotic pathogens, which, as we now know, are very fashionable, but it has not always been the case. And And I do find a challenge trying to convince some of my colleagues that we've got a lot to learn from animals and why pathogens are tolerated in animals, but they cause disease in people. But COVID has actually helped with that a bit, I think. So as you'll all be familiar, we all know what a zoonotic infection is. I don't need to tell you guys about this at all. And of course, there are multiple examples, apart from SARS, which is the one that everybody's talking about at the moment. My passion is really about Salmonella. I've been interested in Salmonella for years, and particularly the species that sit in things like chickens and don't cause disease and jump to humans and do. And when I was digging around for some information about these talks, I hadn't really appreciated that more than 6 in every 10 known infectious diseases are spread from animals. And 3 of every 4 new or emerging infections in people come from animals as well, which will help with my grant applications, frankly. Of course, most of the famous hosts at the moment are bats, birds, and pangolins. And I've spent a lot of my life working with bug boys, I like to call them. They're my bacteriologist friends who have spent years telling me that it only matters, what it matters is what happens in the bug, and that anything to do with zoonosis is nothing to do with the host and only to do with the bug. And hopefully COVID has also managed to persuade some of them that that's not strictly true. So my interest is really in the impact of the immune system, particularly innate immunity. And of course, much of the work that's emerged over the pandemic has shown that bats, for example, have modified type 1 interferon sensing and limited inflammasome activity, which I'll come back to, which is thought to limit antiviral infection. Now, while all this was going on, we had an ongoing project on several species. The 2 types of species I spend a lot of my time working with are dogs and chickens. Probably for me, the game-changing moment that's occurred in the last 5 years is CRISPR-Cas9, because I have always had an interest in comparative biology, but there are no tools. And so as an immunologist, how was I supposed to investigate any of these things if I didn't have, obviously, knockout animals, didn't have knockout cell lines, didn't have antibodies against various cells? And of course, CRISPR changed everything, and we've become quite good at CRISPR, to the point where we can knock out anything so long as we've got a cell line. And I've got a good cell line for dogs and a good cell line for chickens. And I can also tag things. So that means the problems of antibodies not recognizing proteins or cytokines no longer is a problem, because you can just do an ELISA for the tag. So as we were working mostly on the dog cells, sort of early last year, I had a lot of this work good to go, but I was just trying to think, how was I going to sell it to a journal? Because journals tend to go, oh, we're not interested in dogs, who cares? And conveniently at that point, SARS-CoV-2 decided to jump into mink, mutate, and jump back again. And so I slightly, shamelessly, I'm afraid, rebranded my talk around mink. But actually, if I'm honest, most of this work is about dogs, but the principles applied pretty similarly across, we think, and I'll explain why. So back to Salmonella. So Salmonella is the bacterium that I've spent a lot of my time working on, and it's recognized by a number of pattern recognition receptors, which is really my area of top interest. So the lipopolysaccharide in Salmonella is recognized by toll-like receptor 4, and this is one of the primary inflammatory drivers of the Salmonella infection. Whoops. What this does is activate, TLR4 gets activated, it activates the pro-inflammatory transcription factor NF-kappa B, and this then leads to the production of a whole host of Th1-related cytokines, such as TNF. But it also drives the transcription of pro-IL-1 beta and a gene called NLRP3, which is one of the inflammasome-forming genes. Inflammasomes are pattern recognition receptors coupled to effector proteins that are in the cytosol, whereas toll-like receptors are in the cell membrane. And so Salmonella happens to activate at least 2 inflammasomes. Its favorite inflammasome is the NAPE NLRC4 complex, but it also activates the second inflammasome, the NLRP3 inflammasome, which is a fairly ubiquitous inflammasome activated by a lot of activators. When either NLRC4 or NAPE or NLRP3 are activated, they recruit an adaptor protein called ASC. This is a very interesting protein. It forms a massive macromolecular complex in the cell, which is easy to see, and I'll explain why that's important later on. The scaffold then of ASC and the NLRs recruits pro-caspase 1, which is an enzyme that processes pro-IL-1 beta and pro-IL-18 to its mature forms. It also processes a protein called gasdermin D. It chops off its N-terminal. The N-terminal then wedges in the cell membrane, causes the cell to explode and die. And Salmonella can also sometimes activate a protein called Caspase 11 through its LPS. So if LPS gets into the cytosol, as opposed to being retained by the membrane, it will trigger activation of Caspase 11. And this caspase actually predominantly processes gasdermin D and then causes a lot of cell death as a consequence. Caspase 11 is called Caspase 4 and Caspase 5 in humans. And that's important to remember as I go through the talk. So the reason why we were working on this is we were really intrigued by this observation, this beautiful observation in 2008 by Eckhart et al., where they showed that dogs were really weird because they had a very strange Caspase 1, Caspase 11 hybrid. So what's happened is that the normal arrangement of Caspase 1 is that it has a series of card domains, one or more card domains, and a catalytic domain. Okay, Caspase 11 has 2 card domains and 1 catalytic domain. Caspase 1 has 1 card domain and 1 catalytic domain. That's its most simple. And Caspase 1 and Caspase 11 are next to each other in the genome. There's in mice anyway, they're right next to each other in the genome. And so what appears to happen in dogs is that there's been a deletion such that the card domain of Caspase 1 is fused to the catalytic domain of Caspase 11, which is also known as Caspase 4. Okay, so you've got this fusion protein which has got half of Caspase 1 and half of Caspase 11 on it, which is very bizarre. It's not present in any other genera except the carnivores. So when you have a look at the conservation of caspases in carnivores, what you can see is this Caspase 1,4 fusion protein is conserved across them all. Okay. The rest of the caspases are reasonably well conserved between other species. So they've got the traditional this traditional caspases, Caspase 3, Caspase 8, et cetera. But this Caspase 1,4 fusion conserved across everything, and it's only found in carnivores. It's not found anywhere else. So, and this is interesting because you would then predict from this architecture, because it has the catalytic domain of Caspase 11 or Caspase 4, but the card domain of Caspase 1, that functionally you would expect it to behave from a catalytic point of view, like Caspase 11, so to process gasdermin D, but you'd expect it not to process pro-IL-1 beta, okay, because the catalytic site is different. And you'd also expect the function of the card to be like Caspase 1 and not like Caspase 11. So we set out to try and explore that hypothesis. And that's important. The card domain is important because the card domain of Caspase 11 is what recognizes cytosolic LPS. So you would therefore predict that the dog, Caspase 1, Caspase 11 fusion, would not recognize cytosolic LPS, but would process gasdermin D, but probably not process IL-1 beta in response to inflammasome stimuli. So this was before I was really getting CRISPR done in my lab very well, because this is 2016, I guess, 2015, 2016. And at that time in 2016, I was lucky enough to go and do a sabbatical in San Francisco in Genentech with Vishwa Dixit's group. And I had a ball. So, and we did a lot of things there. So apart from learning to bicycle up hills, which if any of you have been to Cambridge, you know, Cambridge is as flat as a pancake, and San Francisco certainly is not. So I had a lot of fun on my bicycle, but we also did a lot of work. And I persuaded them that what would be a really interesting thing to do would be to make a mouse that expressed the Caspase 1, Caspase 11 fusion. And so Soren Warming, who's a fantastic molecular biologist, said, oh, that'd be quite a cool thing to do, because actually we could do something really interesting that's not been done before in mouse transgenics, is we can make a CRISPR-Cas9 deletion of the catalytic domain of Caspase 1 and the card domains of Caspase 11, because the two genes run into each other. So we can actually just lift that whole chunk of the protein out, and you'll be left with, which is exactly here, which is the Caspase 1, Caspase 11 fusion, so directly analogous to what you would see in the dog. So that was groovy, so they did that for me, which was great. And the protein was expressed, and we were all happy. And they were happy because it really was the first time anybody had ever done this, made a fusion protein in vivo. So we then set about to characterize this mouse, which I christened the dogmo. And we took macrophages from this mouse to try and understand then how this protein would signal, and would it signal the way we expected it to. So sure enough, these are macrophages, bone marrow-derived macrophages taken from the mouse, the dogmo mouse. And when we infected these macrophages with Salmonella, what you got was gasdermin D processing. Okay, so this is the cleaved end portion of the gasdermin D. So that was exactly what we expected to see. And just to convince you, this is what you see in wild type macrophages here. Okay, so cleavage of gasdermin D, cell death exactly as expected. So that was great. So then we said, okay, so how does this compare to dog macrophages? Okay, because we expected the dog macrophage has the caspase fusion, should behave the same as the dogmo mouse, and that would all be very neat. But it's not, predictably, not quite that simple. So what I've got here is, let me just see if I can move the pictures. Right, so what I've got here, I need to be able to see the legends, forgive me, is different macrophages, either from normal mice, from the dog cell line, or from two different knockouts. And the take-home message here is this is cell death. And what you can see is that in the wild type macrophages, when you infect them with Salmonella, you get massive amounts of cell death. And this is exactly what I would expect to see. In the dog macrophages, though, it's completely different. So you get some cell death at 24 hours, but you get nothing earlier. And this was really unexpected. I expected this to map onto the cell death we saw in the wild type macrophages. And interestingly, it's also different from the Caspase 1, Caspase 11 double knockout, and from the Salmonella NLR. They're doing something rather unique. And even more surprising, it is actually processing a bit of interleukin-1 beta. So this is the wild type mouse macrophages weren't as enthusiastic as they normally are with producing IL-1 beta. But the dog macrophages here produced quite a lot of interleukin-1 beta, but again, only at 24 hours. And now this maps onto cell death, because you need to have cell death, or certainly pores formed in the cell membrane to let interleukin-1 beta out. So this is very curious. And we sort of thought, well, what actually is going on? So the first experiment we had to do, of course, was look in primary dog macrophages, because cell lines can always be artifactual and who knows what's going on. So we did this. This is the dog cell line, and this is the primary macrophages from the dog. What you can see here is you get cell death in response types, but only at 24 hours. And I can actually tell you this is only when the cell is absolutely stuffed full of bacteria. Interleukin-1 beta, well, the wild type macrophages produced a lot less than the cell line, but there was still a bit of interleukin-1 beta here, again, only at 24 hours. And how does dogmo then compare with the DH82? Well, as you will have guessed from what I've said, the dogmo don't really map on exactly the same as dog macrophages. So we've got wild type macrophages here. What you see here, of course, is the lytic cell death in response to Salmonella. Dogmo macrophages, lytic cell death in response to Salmonella. But really most of this is missing in the DH82 dog macrophage line. Interleukin-1 beta, however, you can see wild type macrophages produce IL-1 beta, interleukin-1 beta. The dogmo macrophages don't, which is exactly what we'd expected, but the actual dog cell line macrophages do produce some interleukin-1 beta. So it is somewhat curious. If, on the other hand, you compare, okay, the activation of caspase 11, so this is where you put LPS into the cell, so you've got cytosolic LPS present. Wild type macrophages with cytosolic LPS, you get lots of cell death, exactly what you'd expect. The dogmo macrophages, you get no cell death. And this is because, if you remember, the card domain of caspase 11 is gone. It's been replaced by the card domain of caspase 1. That can't bind to cytosolic LPS. So therefore, you would not expect to see any activation of the dogmo caspase. And thankfully, at least one effect of dog caspase 1, caspase 4 fusion was conserved between dogmo and dog cells. In dog cells, they also cannot sense cytosolic LPS. So the change in card domain is conserved and does behave exactly as we would predict. So that's pretty interesting. So while I was in San Francisco, I met a tip-top caspase biologist called Guy Salverson, and he does a lot of work with recombinant caspases. So we were chewing this over, and he'd been playing around expressing and making dog caspase 111. So he said, Claire, shall we have a look in our system? And I said, well, that'd be great, because we've got all this data and I'm not quite sure what to do with it. I don't really understand what's going on. And so Guy made recombinant caspase 1.4, caspase 1.11 here from the dog, and he compared its functionality with caspase 11 from the mouse. So this is recombinant protein now, this is not cells. And what he did was he gave substrates to the caspases and measured what happened. And what he found was that the caspase 1.4 fusion from the dog nicely processed gasdermin D, but ironically, it also processed a bit of interleukin-1 beta. Now, the pattern is not exactly the same as we'd expect. When he compared this to caspase 11 from mice, what you could see, again, is that gasdermin D is processed. But actually, the caspase under these situations, when you give caspase 11 enough interleukin-1 beta, it can do some processing as well. So this kind of interesting data here about recombinant caspases, and we're seeing quite an interesting pattern of behavior. So we're going, okay, so what does all this mean? How do I interpret, how do I work out what's going on? So we've got a caspase that's different in the dog, but functionally quite intriguing. So if you know that the recombinant proteins can process the expected substrates, but that in the dog cells, you're getting some processing of gasdermin D, but you're not getting much else, then presumably what that means is that there's some other regulatory mechanism that's important. So that took us back then to looking at NLRP4 and NLRP3, because we thought, okay, maybe there's an upstream effect. So maybe something happens that the NLRs are not very well activated. And the reason why we don't see much activation of the caspase in cells is simply because there's some sort of change in the way these NLRs are activated. And there are some really interesting changes in the dog and in all carnivores, actually, with respect to the NLRs. And this really is very intriguing. So animals from the Canides, so that's dog, dingo, fox, bears, pandas, ferrets, minks, they all miss NLRP4. Okay, it's completely gone, which is really curious. The Felidae miss a protein called NLRP1, which is also kind of curious as well. But NLRP3 is conserved. Okay, so there's very interesting patterns. And the interesting things about the dog loss of NLRP4 and NAPE is actually some of them are pseudogenes. So they're even in the process of being lost over time, but they're definitely not expressed. So we were already seeing patterns of things missing, but this suggested to us that if we've got, in response to Salmonella, if we've got no NLRP4 at all, then surely NLRP3 must be much more important, because it's really important in the mouse along with NLRP4. So we then said, okay, well, we better try and work out what's going on. We'll characterize NLRP3 in the dog cells. Okay, we'll do it clean, we'll do it with the ligand. So we'll take nigericin, which is a cardinal protein that activates NLRP3. It's really super efficient. And we'll kind of do a dose response curve. So this is the wild type mouse cells. You put in nigericin, you get a nice amount of cell lysis, okay? Get a nice amount of interleukin-1 beta, exactly what you'd expect. However, if you put it onto the dog cells, they do virtually nothing. Okay, the only time you get any activation is at 200 micromolar, and I can tell you that 200 micromolar of nigericin just smashes everything to bits, kills everything on the plate, including caspase 1, 11 double knockout cells. So that this is not specific, this is a non-specific effect. So NLRP3 in dogs doesn't seem to be doing very much. And in fact, if you have a look, you can see a trivial, this is from dog cells. You can see this is pro-IL-1 beta in these cells with LPS and nigericin. This is cleaved, this is a tiny amount, efficient. So that was kind of super curious to us, really. Does this then mean that there's no inflammasome activation at all? And the answer to that is no. And the way we were able to determine this was we can measure. Do you remember I told you that the adapt protein ASC, this big complex in the cell, leaks in every species, including chicken. I have no idea why, but this is a phenomenal antibody. And what you can see here is that when we stimulate the cells with nigericin, you can see an ASC spec. And actually, likewise, if we infect cells with Salmonella, we can see an ASC spec. So here's your ASC specs. You can also use something called fluorescence Caspase substrates. So when you do that, you can see a fluorescence Caspase substrate in the same spec as the ASC. And we've used this technique a lot. It's super useful. That's what's going on here. So this is a Caspase 1 substrate, which will also bind inefficiently to the catalytic site of Caspase 11. So you can see there's some staining there, and that's consistent with localization of the catalytic domain of the dog Caspase 1,4 fusion. But what we could also see here was recruitment of Caspase 8. Now we knew, because we'd done this work much earlier, that if you take mouse cells without Caspase 1, Caspase 8 comes into the inflammasome complex to replace it. So this was kind of interesting to us. So, okay, Caspase 8 is there. Wonder what that's doing. And actually, what that is doing is it's processing interleukin-1 beta from the inflammasome. Okay, so you can see here what we've done is we've taken DH82 cells, the dog cells. We've taken out Caspase 8 using CRISPR-Cas9, and in fact, the gene editing was so efficient that we could do this on cells that weren't cloned, because every cell was gene edited, which does happen sometimes. If you take those cells and you infect them with Salmonella, what you can see is a complete loss of the interleukin-1 beta in these cells in response to infection, okay, compared to the electroporated control cells. Cell death is untouched, the delayed mass non-Caspase 1 driven cell death. So Caspase 8 is not contributing to this process, but the interleukin-1 beta is actually being processed by Caspase 8. And you can see this here, it's really neat. Okay, so this is pro-IL-1 beta, this is cleaved IL-1 beta in the wild type cells. And if you take out Caspase 8 or its upstream, or its upstream effector protein, RIPK1, there's a loss of any processing of IL-1 beta. And so this is kind of intriguing because Caspase 8 is super important in a number of cell death pathways. And so we're now thinking, there's something really complicated here with the cell death pathways going on. The dog cells are not dying very efficiently. We've got some role for Caspase 8 in processing IL-1 beta, but what's happening to cell death, we don't really understand. And the reason why it's complicated is because there's a lot of overlap between different cell death pathways. So we knew that normally you'd expect NLRP3 to activate Caspase 1 to drive cell death, okay? But we weren't really seeing, we were certainly not seeing any cell lysis, minimal cell lysis in these cells at the time points when inflammasomes are activated. We're seeing it after 24 hours when the cell is completely stuffed with bugs, but we're not seeing it any earlier. But what we were seeing was some Caspase 8 processing of interleukin-1 beta, which presumably is coming by Caspase 8 being recruited to the inflammasome. But we weren't really sure what other cell death pathways may be involved. And critically, the dog is particularly peculiar because there's another important lytic cell death pathway, which is driven by some process called necroptosis. And this is dependent on an effector protein called MLKL. And like gasdermin D, MLKL goes to the cell membrane, forms pores, and causes the cells to lyse. But dogs don't have MLKL. And in fact, carnivores don't have MLKL either. So this is super curious because that means that you're that carnivores are lacking 2 of the really important lytic cell death pathways. And in fact, the only way the dog cells are dying is by a mass action effect where there are so many bacteria within the cell that eventually it just lies because the membrane can't contain it anymore. So that's kind of curious, particularly because both pyroptosis and necroptosis are supposed to be super important host protection mechanisms. Okay, so it's really very, very interesting. So we did one final set of experiments to see, well, is there any kind of activation of gasdermin D? Are we just getting a little bit of it being activated to form holes in the membrane, but that the cells don't rupture? Because there's nice evidence in the literature to suggest that if you get a low-grade activation of inflammasomes, you can get a few pores formed, but the cells don't lyse and die. And in fact, this is exactly what we saw. So this time we used a technique called Protium iodide uptake, which is where you put Protium iodide in the media, holes form in the membrane, it gets taken up, and then it fluoresces when it's inside the cell. And so we did see that. We would see in response to the very high dose of nigericin, you could see that eventually some dye went in. In response to salmonella infection at high multiplicities of infection, some of the dye went in. The rest of the time it was pretty non-specific. So overall then, inflammasome activation in the dog and presumably other carnivores is frankly at best weedy. So that's kind of intriguing. We were left with data, of course, that we knitted together for the paper that's published in Cell Reports. We saw these big holes in the pathway, but this also paralleled another paper that we did with our colleagues in Australia, where we looked at mouse control of salmonella. And what we found that the only way that a mouse can lose its ability to control salmonella is to lose all its cell death pathways. So if you take out pyroptosis, necroptosis, and apoptosis in the mouse can no longer control salmonella. But in the dog, without pyroptosis or necroptosis, you're getting really strange control of salmonella. The dog cells can still apoptose. But we're there for some really interesting questions, because what does that actually mean? What does that mean for the carriage of bugs? What does that mean for the carriage of pathogens, zoonotic pathogens? How does it all work? And so obviously we can't answer those kind of global questions. So we were left with some conclusions about this work, which was that inflammasome activity is very much dampened then in carnivores, including animals like mink, of course. Lytic cell death markedly reduced. There is the capacity to form some pores, but it's not really leading to cell lysis in response to inflammasome activation. The cells can't necroptose because MLKL, the gene, is gone. We did wonder, there was some really neat work that some of our ecology colleagues came to present that showed that if you had a very high protein diet, that this was done with maggots, actually, it's very interesting. The maggots produce lots of antimicrobial molecules. And so we thought, well, maybe that's what's happening. Carnivores are obligate to eat meat, obligated to have a high protein diet. So maybe they just don't need these genes because they've got antimicrobial peptides being produced in the gut because they've got lots of protein that allows them to control pathogens that are there, and it just is not an issue to them. I don't know. But we then reasoned that this could then be a problem, because perhaps then this allows them to carry things and not express disease in a way that we wouldn't have predicted, and hence the reason why potentially they can carry zoonotic pathogens. And actually carnivores can carry more zoonotic pathogens than bats, but nobody seems to talk about that. And then just to further the intrigue, certainly from my point of view of cell death, which is something I'm super interested in now. We've had an extensive program of work working on chickens, and this isn't published yet, but I will cut to the chase and tell you the story in chickens as well. So chickens have a bizarre lack of inflammasome genes. They have no NLRC4, they have no NAPE, they have no NLRP1. In fact, the only NLR they have is the NLRP3 homolog, which is called NLRP. They don't have AIM2. They don't have Caspases 11, 4, or 5. They don't have gasdermin D. They have pro-IL-1 beta, but it's not cleaved by caspase 1 because it doesn't have a caspase cleavage site. So the potential program, and the chicken macrophages do die in response to salmonella. 50% of the cell death in response to salmonella is driven by necroptosis, because they do have necroptosis. Presumably the rest is driven by apoptosis because they certainly don't have the capacity to induce gasdermin D driven pyroptosis because they don't have gasdermin D. And or alternatively, perhaps it's driven by the lysis of a protein called gasdermin E, which is another gasdermin, which has been shown to be activated in certain apoptotic pathways under specific conditions. So we're kind of trying to dig that out at the moment, but it's very intriguing. So basically, I started out saying to you that is cell death a conserved pathway and is it important in zoonosis? Well, cell death is sort of conserved, but only sort of, and it depends upon the circumstances under which you find it. And it seems to be very different depending upon what animal you look at. So it reassures me that mice are useless because they don't really reflect anything useful at all. And there's a lot to be gained, I think. And with CRISPR-Cas9, we do now have the technology to get at some of these questions as long as we have reasonable source of cells, which is kind of the thought I'd like to leave you with. And these are all the fantastic people that have helped do this work. On the left are the guys in my lab who either generated the knockouts or did all the cell biology work. And on the right, the evolutionary biologists in Cambridge, Lucy Weiner, is fantastic. At Genentech, Soren and Nobu really helped me get the mouse done. And of course, this was all under Vishwa's help and guidance, who is just a really fantastic individual. And then Guy Salverson in San Diego at the Sanford Burnham Institute then helped us with the recombinant work. And I have to always thank my funders, Wellcome Trust, Genentech, GSK, and Northera all contributed to funding this work. And I'd be delighted to take any questions if you have any.
Wonderful. Thank you so much. So the floor is open for some questions.
So fascinating talk. It's interesting, as I alluded to. We've been doing some of the modeling of the bats with respect to differential interferon, dynamics, and also this inflammasome issue.
Mm-hmm.
So if you don't mind, I'm asking a couple of questions.
Sure.
So one is, are there differential disease response phenotypes in carnivores for pathogens other than Salmonella? Right.
So that's kind of an interesting question for a veterinarian, right? So I thought about this a lot. Having seen this, I was really intrigued by that. So yes is the answer. So cats and dogs, they're certainly peculiar with respect to enteric pathogens, right? So anybody who's a clinical veterinarian will tell you that the most tedious thing you see all the time is diarrhea in dogs. Okay, so dogs are hugely efficient at getting rid of pathogens because their net zero response is to expel them all with diarrhea. We don't see much bacterial translocation across the gut to cause any kind of systemic disease. And in fact, cats are even more efficient they're not actually more efficient at diarrhea. I'm sorry, this is kind of a revolting conversation.
Okay. I'm a surgeon, so I listen.
Oh, that's fine, that's fine. You'll understand that. Not everybody understands, right? But cats seem to not sense a lot of bacterial molecules, so they have wacky responses to LPS, for example. You need enormous doses of LPS. So to give you an example, right, a cat can have a chest full of pus. I mean, and it should be seriously sick. The only thing it cares about is it can't eat. Okay, so there is actually quite an interesting species differential in the response to certainly bacterial pathogens. Viruses, of course, viruses are unique compared to each species. As you know, each species will do something different. So it's a little bit hard. It's a little bit hard for me to trot across because the viral adaptation seems to be more efficient than bacterial adaptation. But I can certainly tell you that enteric pathogens and the cat response to infection is completely different.
Yeah, so I think I think that's one of the interests in NLRP is the primary, the unknown responder, right? The more ubiquitous responder for inflammasome activation. There are going to probably be these differential evolutionary adaptations with respect to other compartments of inflammasome activation, as you talked about, that are specific to the environment and the diet and whatever of different animals, et cetera. So I think that this this parsing of the differential forms of cell death is incredibly important with respect to the manifestation of disease, since the pyroptosis and the necroptosis are pro-inflammatory death forms.
Yeah, absolutely. I agree. Yeah, I agree.
Yeah. So my other question about this was, trying to remember what that was. Oh, so in one of the findings from our computational modeling of bats is that if you represent these kind of differential inflammasome and interferon dynamics, you can get some separation in terms of the disease manifestations phenotype, but it's really not that great. I mean, it's not to the degree in which we're seeing bats being essentially non-diseased carrying these viruses, whereas humans, have the potential to get quite ill.
Yep.
And so we're looking.
Carnivores has joined us, I'm afraid.
So we've been looking at and thinking about inflammasome properties in other cells other than macrophages per se. And we're particularly interested in endothelial inflammasome activation as an accelerant of localized effective antimicrobial function and the expansion to disadvantageous systemic sort of thing. So have you looked at inflammasome properties in different endothelial cells in your species, or?
No, I'll tell you why, because we haven't got an endothelial cell line. So that then makes life somewhat challenging. What I can tell you is there are very interesting differences in epithelial cells, okay? And this is again something the mouse boys won't talk about, but actually NLRC4 and Caspase 11 are really important in the mouse gut cells. And the idea is that what happens is the cells die in response to an infection that takes them out of the gut, and so therefore you don't get the bacteria crossing cells. Yeah, so, which is cool. But then if you ask people about humans, okay, and eventually some of the NLRC4 people will admit that actually there's incredibly low levels of NLRC4 expression in human gut, okay, and that they're kind of really willing, nobody's sort of, they're sitting on the fence a bit, right? But I suspect that NLRC4, possibly Caspase 11, but I'm not sure, but certainly NLRC4 in the epithelium is a mouse-specific phenomenon, and I suspect that's not important for other species at all. And the endothelium, I used to work on the endothelium many years ago, and it's super, super, super interesting, but I don't have an answer because I don't have a cell line. If I had a cell line, I'd do it, because I think I'm a little macrophage obsessed, obviously, but I'm seriously interested in sepsis and sepsis syndromes. It's immensely important in some species, but it's not important in other species. Cats and dogs, cats not at all, dogs minimally, horses massively important, right? And if I could get a hold of endothelial cell lines, that's something I would do, because I think that's a really interesting question.
You can't get them out of your chickens? You can't grow endothelial cells out of your chickens?
I probably could, if I thought about doing it properly. Yes, I think that's probably worth a shot, actually. I hadn't really got that far, but actually, I think that's something I do need to give some consideration to. I think you're right, because ultimately, because you see, the other thing I'm thinking about is I don't know, you might know the answer to this, because I don't know the answer to this. I don't know how efficient it is to grow these things from iPSCs. So we can get macrophages out of iPSCs, and it's fairly easy to generate iPSCs from different animals with a little bit of tweaking and cloning of growth factors and so forth. So I don't know whether that sort of strikes me as the other thing to do, right, which is to generate iPSCs from the different species and then start to generate the different lines that you need, because that's probably the other easy way to do it. But easy in inverted commas. I mean, we've done a lot of work with iPSC to macrophage generation. There's nothing easy about it, but it's tractable.
I wonder if you can get endothelial precursors from eggs. Because a common source for humans is human umbilical vein endothelial cells that are.
And you know what? It's really easy to get. That's actually a really good idea, because it's really easy to take the vessels off the surface of an egg yolk. So that's a really good idea. I hadn't thought of that. Cool. This is why I like doing talks.
Well, if you end up doing that, let me know that you've done it, so that we would model that. I think. And I don't want to, more monopolize the questions, but the chicken thing is really interesting because one of the things that we're thinking about is a final common pathway disease mitigation strategy, which is immunomodulation, right? Because if you focus on the viruses, there are a million viruses, you have to come up with something new. Are there common pathways towards disease manifestation? Because all these respiratory viruses share the same sort of pathophysiology, which is inordinate manifestation of inflammatory function. And that very likely has some endothelial component associated with it. But, so the question is, from a comparative biology cross-species system, right? Whether or not we can gain information about things that confer disease resistance or not, right? In organisms that have been known to be significant human pandemic zoonotic sources. So you mentioned that people aren't too concerned about, bugs coming from carnivores. I think it's because we haven't seen too many pandemics coming from bugs that come from carnivores. So I think that there's something about the the evolutionary inflammasome and inflammation adaptation associated with these pandemic generative species that somehow leads to viruses coming from those species having high pathophysiologic potential in humans. And I think it would be interesting to think about comparative, doing some sort of comparative biology focused on pathophysiologic potential arising from how those particular organisms have adapted to potential transfer of viruses.
Yeah, that'd be great, actually. And so we should probably continue this discussion between ourselves, really, instead. But cell death, I think, is central to that. Right, I think you can arrive at it with different solutions, but the long and the short of it is, having spent years trying to work out. I spent years of my life trying to work out what PRRs are important, and I've suddenly, I sort of had this epiphany when I was doing all this work and thinking, do you know what, Claire, everything dies.
And how you die matters. How you die matters.
Dying matters, and all the PRR pathways end up in cell death, right? So we should talk some more. I think that'd be very interesting. I'm really interested in that.
I'd be interested also because this issue of the control of cell death is so important, and people, for so many years, people said, "Oh, there's apoptosis on one hand and necrosis on the other," without the appreciation of the complexity of the regulation of cell death and the effects. And in toxicology, when cells in your liver die, it really matters how they die.
Yeah, of course.
And a lot of the downstream pathways, the effectors at the top are different. But those downstream effectors and the way the cells die are the same ones that you're mapping out here. And those intermediate layers are the same. The Caspase layers are the same. And so I think that it's, and the comparative study is critical. I mean, my one comment to Gary is maybe we don't see some of the carnivores as sources of zoonotic epidemics, not because they aren't sources, but because we've been exposed to them so regularly and so long that those are diseases that are generally diffused. Whereas with bats, maybe the interactions are rarer, so we run into that we see them as new diseases more.
Yeah, that's possible. Chickens and pigs, we're around them a lot. We get sick from them.
But that was fascinating. I'm sorry, I don't want to cut you off because it's such an incredible conversation. Is there anyone else who has a question? Poor Reinhard has been sitting here patiently waiting to give his presentation.
Yes, I had a question.
Please, Ruchira.
Yes, so thank you. That's for the fascinating talk. So this is about the one thing you mentioned from Carter et al. So it's about the diet perhaps modulating the immune expression. And so since you said that, I just pulled up the paper and barely tried to look at the abstract and introduction, and it seemed that they were seeing that as due to the nutrient content, but I was wondering if someone had looked at the fact that the microbiota on the food itself would be different, and that would have a different effect on the microbiome, not just the pathogenic microbes, but the whole the general gut microbiome.
And of course you're right, and the answer is the work that when we quoted it was fairly early on in the scheme of things, and it to be honest, it wasn't something that I'd even thought about until we started to see these results and we were trying to work out what was going on. So I don't know how much more work has been done on that, but I think it's obviously super useful thing to do. Right, I'd be really intrigued to understand the differences in the microbiome and how that then tallies with what we're looking at and the interaction of that with the gut immune system. But the honest answer is, as far as I know, not much more has been done on that. I think it's an open game, right? To be frank.
Okay, thank you. Yeah, lots to find out.
Okay, well, we promised we'd let you get some rest. So I want to thank you again for a wonderful talk. I hope maybe you'll be willing to consider coming back and giving us another talk, because there's so much material there. You could easily give a series.
I'll be delighted. And I would be delighted to talk to you and Gary in a separate meeting so we can talk about this further, because it's it's very exciting. So
I'll shoot you guys an email. We'll set something up.
Okay. Fantastic.
Thank you so much.
Thank you everybody for listening.
Thank you.
Bye-bye.
So Reinhard, thank you for your patience. I'll turn the floor over to you.
Thank you. I have decidedly less exciting things to tell you, so there's no need to interrupt these interesting discussions. What I want to do is give one of those periodic talks we have from the subgroup. So for those of you who are not regulars, this working group has various subgroups where a good part of the work gets done. And the subgroup I'm talking about is, let me maybe share my screen. How can I share my screen? Here we go. Can you see my screen?
Yes.
Maybe to presentation mode. All right, there we go. So, so the subgroup I'm going to tell you about is the subgroup on crowdsourcing models. And you see here the focus of this group. It was established by Paul Macklin and myself, and it really came out of a particular view of models that I have and that Paul has and many others, I think, in this working group. And that is, for this particular topic, it is models that have many different components. We of course, this group primarily looks at the immune response to various pathogens. And so the kinds of models that I think are in part relevant are models that are built from a number of different components. And in the last year or two, there were several community efforts to build larger models by putting together component models. There was the COVID disease map built by a large European consortium. There was Paul Macklin's effort around COVID. There was James Glazier's and others' efforts around this. And the idea was, you know, first of all, what can we learn from these efforts about what does it take, what sort of social and technical infrastructure does it take to build such models, and how can we leverage that to build others. And so that's what this subgroup was born out of. So what did we do? Like most of the other subgroups, we had discussions about all sorts of things. Various obstacles to sharing models. So these are things that have been discussed in this community for a long time: reproducibility of models or lack thereof. How do you share models? How can you adapt models for other purposes? I think the consensus more or less was that it happens rarely that models are adapted or reused even, which has a variety of reasons. What are obstacles to collaborating on model building and integration of models? And what can we learn from existing efforts? We put together a whole long set of notes that we're happy to share that collect some of these thoughts together. And eventually what we did is we refocused a bit on immune digital twins. That was one of the themes that emerged over the last year in this working group. And we turned our efforts to turning our set of notes into a manuscript to be published. Now, when I say digital twins, there are many different concepts and definitions of what digital twins are. It could be a machine learning algorithm. It could be, simply a differential equations model, or it could be something much more complex. We have a particular focus in this subgroup as an immune digital twin as being a complex model that's multi-scale, involves a number of different component models, both at all scales, intracellular, tissue, whole organ. And so we're almost at a point where we have this manuscript done. Here is the title, at least the working title. And this is the list of authors currently listed in alphabetical order. And the idea is to submit this as a prospective article of some sort to a journal, and we haven't entirely settled on what journal that will be. So we'll see. Now, what's in this paper? Very briefly, it's a description of what we think of a digital twin and what we consider the steps to build one. So, and this is really modeled after the sort of the classical digital twin concept that comes from industry, where you have some machine or mechanical device or a system that is built based on essentially a mathematical specification, and then it is individualized to a particular instance of this machinery through data taken, typically operational data taken from that machinery. And so step one would be to build such a generic template model, in this case of some appropriate portion of the immune system that would be relevant for a particular chosen application. So obviously it's not feasible to build a mathematical model of the immune system. And that's not the concept of a digital twin either. It has a specific application in mind. So that's step one, this generic template model. Step two is to personalize this to an individual patient. So this could be done in a number of different ways. So it might be done essentially online. If you monitor a patient, you get streaming data, and you want to adjust, for example, how this patient is treated over time. Or it could be what one could call offline. That, for example, if you try to decide on, for example, a particular procedure to repair a blocked artery based on imaging of the individual patient's heart. And then the third and most important, the most difficult and probably most expensive step in this whole procedure is to actually validate this immune digital twin so that one can trust it to be used. There are many unsolved issues related to that. In particular, depending on the application, it might be difficult or impossible to get the appropriate kinds of data. For example, if you care about lung immunity, which we do, then you need data from a patient's lungs rather than a patient's blood, for example. So these are the basic steps. Here's an image from the paper that sort of gives a schematic outline of how these different steps are that go into it. So you have all the different scales, all the way from the molecular to the whole organism level. You get data from that, you integrate them into a model. You add data from a particular patient, operational or particular data, and then the resulting model you go on and use for whatever purpose you have to use it. Trying to advance here. There we go. This is simply a diagram of the various steps that go into it. Not very exciting, but these are some of the nuts and bolts that you would use. The paper ends with a collection of action items. I'm not going to go through those. These are some of them. There are more. Essentially, you need to get started in figuring out, what goes into this. What is the conceptual map of an immune system component for a particular application? Once you have that, how do you actually model it? Who is going to contribute? How is the whole thing going to be funded? And then there's the whole thorny issue of standards, data management. How do you make this transparent and replicable? And depending on what your end product should be, for example, if it is going to be a medical device, you will need to have all the documentation required to get approval. So in a nutshell, that's the paper. If you're interested in a draft, I'd be happy to share one. Maybe not this current version, but the next one. And so then we've had quite a bit of discussion as to what is this subgroup going on to do next. In some sense, I think the subgroup has achieved its purpose in learning from existing efforts, figuring out how those can be leveraged to build large-scale models focused on the specific digital twin concept. So one might consider that here with the life of this working group is coming to an end. And there were some discussions about whether it might make sense to close this subgroup and start a new one that actually focuses on the topic of this paper, and that is on the actual building of the digital twin. There's still ongoing discussions. The question is, what really would be the focus of this working group? What would be the output? What would be the end product? If anybody has any good ideas of what one could do that would be productive, then I'd love to hear them. So that's really all I have to say. Any questions or comments are welcome. Maybe I'll stop sharing here.
So I'll let you steer, Reinhardt.
Yes, I'm happy to answer any questions, but in particular, I'd be very interested in some thoughts you have about where one could take this now, right? Essentially, we've laid out at a high level, admittedly, what we think needs to be done, and the obvious next step is to actually do it. But so what does that take? Can a group of people here make a difference, do something productive, or not? So please.
Yeah, so I think I'll read. I don't know if you had a comment. I just saw you comment.
Yeah, I had a quick comment. That was really interesting. Thanks for showing that. I think I'd be very interested in a copy of a manuscript whenever it's available. Keep it confidential, sure. Obviously, until it's submitted. Yeah, I think I've learned a lot about digital twins. Like the concept of digital twins is gaining interest across NIH, obviously, and there's a lot of internal conversations there. I mean, I think one thing you highlighted in that talk, and I get nervous saying things and then not being able to follow up, but we do have provisional approval for through NIAID to have a meeting in late 2022, or I'm sorry, late 2021, around multi-scale modeling of pandemics. And it's so we have limited funds to do something like that. It wouldn't be necessarily in lockstep with the IMAG. However, it could be. And perhaps, I think it's just something to keep in mind. We're still kicking around ideas, but perhaps like this could be an element of that in a public forum. Maybe the timing will work out where your paper is submitted already, and it's something to consider. So those are my two comments. I do like the way you've outlined it, but the figures you showed are easy to digest and understand and might resonate with folks here. So I think those are my two comments.
Yeah, that's great. Thank you. We hope to have this submitted by the end of the year. It's one of those items on our to-do list to get this off the books by the end of the year. And I mean, certainly as you saw, the first item on that to-do list is to bring together people to figure out, how can one do this? What needs to be done? There's a number of different approaches. Digital twins could be used for different purposes. They could be used for, say, creating synthetic populations, various other approaches. And depending on what your intended use is, some of these procedures might need to be done differently. But I think it for starters, needs a lot of discussion among modelers, among immunologists, and among clinicians. I think you need to bring together those three groups. So if there's an opportunity to have such meetings, I certainly would be extremely interested.
Yeah, that sounds good. I mean, I think let's see where it takes. I mean, we have, like I said, we have approval for this one meeting, and it would, I don't know what the schedules would be, but it would be later in the year. So perhaps take it a step down. I'd love to see the manuscript when it's ready, and then maybe we could talk about how to incorporate. It might not be just about that topic, but maybe an element of that meeting.
Sure. Great, thank you. Ruchira.
Yes, thanks. This is a interesting proposal, interesting idea. So I had just put in the chat one paper from the Integrated Mathematical Oncology Group. I mean, they have lots of work on what is roughly digital twins, personalized therapies. And this is one paper referring to that the immune response is important for personalizing cancer chemotherapy. Mm-hmm. So that seems like that's sort of, I mean, they've been doing that kind of thing for many years, and that's obviously has a layer removed of complexity because it's a non-transmissible disease. So I was wondering if we could somehow leverage their experience or and see and then put that together with the network science that is, or the epidemiology that is necessary for transmissible diseases. If we can see how can we slot in the experience of people who are working on a non-infectious disease and have done a lot actually on medical digital twins.
Mm-hmm. Yeah, absolutely. I mean, Moffitt is just down the street from us, and I know the people there quite well who have worked on it. So that's definitely a place to go and learn from their experience for sure. I think they've worked quite a bit on creating synthetic populations for clinical trials and so on. Yeah.
So maybe some of them could give a talk. I don't know who's the most.
So we could ask David or Sandy to speak.
Yeah, David Basanta or Sandy Anderson. Yeah.
Asya, Heiko, there. I mean, there's so many good people at Moffitt.
Mm-hmm. Yeah. So there's a question in the chat by Leonard. How can you join the subgroup? So at this point, there doesn't exist one yet. As I said, we're still discussing. Ultimately, what would the charge of the subgroup be? What should the subgroup be? Of course, it could just be, a discussion group, which is interesting. In fact, I think my experience with this working group and the subgroups and the steering group has been that it's always been a fascinating opportunity to hear a lot of smart people give their opinion on a lot of interesting things. So that certainly is one possible thing to do, is to simply have discussions on different aspects of this topic. I think I generally feel that the most productive way of doing this is to have a specific focus and a specific deliverable at the end to focus discussions. And that's what we're still looking for. But if there will be a new subgroup, then certainly we'll announce it to every member of this working group, and then there'll be a way to join. Okay, if there are no more questions, then I appreciate it. Thanks for staying on late.
So, last chance for questions or comments. People could talk about, even though Clare left, people could keep talking about Clare's discussion too. I mean, the overlap between the information you need to build immune digital twins and the experiments Clare was doing was very high. So, in a sense, I think one of the problems you're going to face doing digital twins is trying to extrapolate from animals where you could do detailed experiments to humans where you can't. And I think her talk was a very salutary warning on how different animal immune responses are from human. And as she pointed out, that those differences are not relevant only to infectious disease, because if caspase and downstream activators are different, cancer responses will be different, for example. And so, as I was saying, toxicology responses will be different. And therefore, that extrapolation is going to be even more challenging than it might have been. At some level, we know that extrapolating from mice to humans is difficult. But to see that kind of molecular discussion of it, I thought was really intriguing. But I think it's not a counsel of despair. I think it's actually an argument that the kind of modeling, maybe not a digital twin, but at least a digital immune system, which you could explore in detail, quantitative predictive immunology using modeling, that you could ask the question, what is the difference that you expect when you knock out pyroptosis, when you knock out TLR4, when you knock out a caspase pathway? Those are kinds of questions that you could explore, I think, very effectively in modeling, that may not be quite digital twin. They might be more of what I'd call virtual tissue, but nevertheless could be very, very important in trying to put these ideas together. And so I think actually there's more commonality between the two presentations today than we might have expected.
I mean, this whole issue from, extrapolating from mice to humans is an important issue, because this template model that you build for any part of the immune system, if you want to do any kind of validation, a lot of it will have to be done using animal models, in particular mouse models. And then you're faced with this problem, how you would transfer this model to say something about humans.
I guess one thing that was interesting was how different the species that she was looking at were. And so even if you say mice are not humans, dogs are not humans, cats are not humans, if you repeat your experiments in dogs, mice, and cats, that variation between them might give you some insight into the space of possible responses that could be relevant to humans. Gary might jump down my throat on that, but I thought that those pathway diagrams and actually the molecular structure diagrams that she was showing were really informative, trying to understand how the space of possible immune responses could look across species.
Yeah, I mean, I think that is, I think, yeah, so that's interesting. So clearly that's interesting. I think one of the challenge is to have some process by which you identify what is actually conserved, because we always know what is different, right? And to be able to parse what the contribution is of what we consider to be conserved. So to go to the digital twin, the conceptual model of specification of the immune system that you're going to lay out is a statement of what you believe is being conserved across either individuals or across species if you're going to try and represent the immune response across those different species. Right? That's what, you know, my editorial in BMB is all about, is that the idea of using multi-scale computational models as little t theories that represent what is conserved from one circumstance to another circumstance. And then the question is how you operate over that. We have developed a whole series of methods to do that, but I'm not saying that that's the only way to do that, but this is a component of a fundamental question of how do you actually translate knowledge from one biological context to the other biological context, you know. And I've made the statement before that the challenge is that without being constrained by known fundamental natural laws that can represent a first principles standpoint, you know, it's hard to do that. And we're dealing with epistemic uncertainty. We have to deal with, you know, a whole series of things that are challenging to examining biology. And I think I would hope that having stated this particular problem, people would be interested in trying to develop sets of robust and generalizable approaches in which you can actually do what we would like things to do. You know, and any of us who have dealt with experimentalists, if you look at the error bars, even among genetically identical mice who are ostensibly supposed to be the same and have the same response, they do not have the same response. So there is some sort of, you know, threshold in which, you know, this is aside from the point. But the fact is that there are these fundamental issues with respect to how to deal with what is a sufficient level of representation of a biological system, you know, to be able to do that. And it underlies digital twins, it underlines comparative biology, it underlines virtually everything that we talk about with respect to biology. I think, you know, so to be more concrete with respect to what Reinhardt was talking about, I think a working group that would be in some ways structured along the way that the old IMAG 10 simple rules, right, for reproducibility, with a very targeted sort of goal, which might be what is the component organizational structure necessary to be developed for a digital twin to be developed, right? What are the components of that? What are the actual tangible objects, right? Be they computational or organizational or whatever, to be able to do the things that you need to do, right? And then somehow make a distinction between just a model repository, right? A whatever, you know, that is just a set of words or a consortium or whatever. Because what we want is we want a tangible object, right? So to move towards that, we have to look at some of the successes and the failures of other attempts to try and create these consortia type models, like the virtual physiologic human, you know, which is good for certain things, but is completely useless for a whole bunch of other types of things. You know, these model repositories, which are useful, but as we noted before, no one uses anyone else's model. They make their own, right? So they're and the issues of interoperability and all that type of stuff, which is laid out in the paper that, you know, that Reinhardt talked about. But turning those particular concepts into a tangible organizational object, right, that can produce an actual thing that represents a digital twin in whatever form that you just specified. I think that is a worthy goal of a working group like that, because then there's an actual output from it.
William, can you elaborate a little bit on your comment you just put in the chat? I think it's quite interesting.
Okay, I can try. It's kind of half formed, right? But I was just thinking, you know, Gary was talking about, you know, thinking about what might be conserved between between different models of different species or that sort of thing, right? So I was just thinking, suppose you got a really good model of a mouse, right? And you can talk about what is conserved when we go from that to a model of, I don't know, a horse. We can talk about what's conserved when we go from that to a model of a dog, and those might be different things, right? I mean, there'll probably be some overlap, but they won't keep exactly the same things, right? Then go one step further. Now, okay, we've got our model of a horse. It's got its stuff in it. We've got a model of a what did I say? A dog? I don't know, a cat, some other thing. It's got some stuff in it, right? And we want to talk about the relationship of those models to a model of a human. So what is kept through that process of transforming from a model of a mouse to a model of a dog to a human, and from the other path, from a model of a mouse to a model of a horse to a human, for example? So if we're talking about these sort of models as objects, and we have relationships between them where some information is lost and some is kept and some things are added and some things are taken away, and we think of this space of models, and we can think of the relationships at various steps along. And I don't know exactly what that gets us, but I don't know. That's my half-formed thought, anyways.
It's a phylogenetic model tree.
Kind of, yeah.
Well, so I think, William, what you're saying, I mean, I like to separate this concept of model structure from parameters.
Sure.
But in fact, there's, to some extent, there it's a fiat distinction. It's a convenience distinction, because you could imagine a neural network which has all-to-all connections, and then you have a lot of zeros on the connection strengths.
Sure.
So you could imagine building a model which has every possible connection. But of course, one of the things that we're seeing today is that there are, in fact, elements of the immune response that are present in some animals and not in others, in subspecies and not in others. And more interestingly, there are molecules that mix pieces of molecules from others, so that the pathway interactions are in fact different. The way signals are transduced and mixed is different between species. And those, to me, are fundamentally structural, model structure differences rather than parametric differences.
Well, yeah, so we can debate this particular issue right till the cows come home. I think part of the challenge of biology, right, is that there are all these potential imputable differences. You know, the thing that makes biology hard is that we have no way of establishing what is a conserved representational structure that can represent a wide range of differential phenotypes, right? So I think that is because we have no equivalent to, laws of motion or conservation laws or any of those other types of things. So building a phylogenetic tree in which things are potentially different, is incredibly important, right? If you're interested about that particular level of detail in parsing something out. There is another, and that's a kind of I think of it as a watchmaker scientific approach, where you want to know more and more about this type of thing. There's another scientific approach that is so that's like the infinity between 0 and 1. Then there's this infinity from 0 to infinity, which is an expansive sort of idea about, well, how many things can I sufficiently describe, right, with as parsimonious a model as I can come up with. And I think that is kind of what has been the tradition of a lot of mathematical biology, with the downside that those models have very little utility because they don't represent certain key biological properties like heterogeneity, stochasticity, or whatever, right? So finding this kind of middle ground between highly abstract, the SIR model is a primary example of that, right? At a very macroscopic level, it's a great model, but the minute you try and use it, it can't use it, right? So I think that finding some sort of operational level And this goes again to the digital twin, because this is defined by the use. The intended use of the model should drive the level of abstraction of the model that you're intending to do, right? So as a clinician, I'm interested in control. I am interested in controlling these systems to be able to move them in the direction I want them to be able to be moved, and with the sets of resolution that I have. And therefore you come up with, levels of representation that suit that. So I think recognizing that there are these multiple tiers of abstraction and representation that are based on use, I think is an important thing. So again, to use another example, physical models of hemodynamics and cardiac performance are very simple, right? Have no molecular detail and are extraordinarily useful and translatable across any sort of animal that happens to have a pumping heart, right? So, that the form of that particular equation that represents that function is conserved across many biological contexts, right? And can be differentially parameterized to provide the equivalent level of resolution control, i.e. changing contractility or changing the volume status, right? Irrespective of what the organism happens to be, as long as you sufficiently parameterize it, right? So the golden age of physiology produced all these elegant mathematical models that have this kind of descriptive and translational power. The problem now is that we are now operating in a control space in which those assumptions that underlie those particular models do not hold. And the challenge is coming up with appropriate scale representation or abstraction level representation for the particular task that you actually want. And so I think that's like an almost an orthogonal axis, right, with respect to the translational things that William was talking about, because that is, breadth of representation across different types of organismal phenotypes. But then there's another perspective with respect to the frame in which that phenotype is actually represented.
Oh, sure. No, I completely agree. I mean, you have, I mean, one thing that it seems like it would be a good idea to do for this kind of working group that was proposed is, well, I mean, Reinhard said it though, but is come up with a concrete goal, a concrete use case, because you're right, that will drive, that will drive the levels of abstraction that you need. And, but I think also, I mean, I was thinking just because of the discussion that was going on earlier, this translating between different organisms, but I mean, you also have, there are relationships between models at different levels of detail of the same thing for the same organism, right? And
that's the principle of experimental biology, right? The justification for studying macrophage function in a mouse is the supposition that it is similar enough to macrophage function in a human such that that information is translatable, right? We know that that's actually not true at a parametric level, right? But the supposition of the entire endeavor is a supposition that the conceptual relationship model established has some information in terms of how the human macrophage actually works. Right. I mean, the links between comparative biology and experimental biology, they're almost completely opposite, right? Because in terms of experimental biology, they kind of don't want to think about the inability to actually make that translation, whereas comparative biology is focused on the differences in that particular translation. Right, so somehow melding those two things is required, because we talked about it, a second ago with respect to trying to calibrate models to in vitro systems. We know in vitro systems, in terms of like parameters, have nothing to do with how those systems actually work in vivo. Right? Sure. And yet we make a supposition that the model structure that is obtained from that in vitro study is somehow conserved when we, operate on that in vivo context. All right. Well, maybe we can get back to Reinhardt about some, some more concrete or tangible targets for what this digital twin thing should look like. And I think that would be a first action item for the working group, is to come up with what that concrete structure might be.
Yeah.
Well, I'm glad that, I'm glad that Reinhardt got to present briefly, and we had such a lively response. And I think that's great. We can maybe have him come back and give the first talk of the day, and then we can have a longer discussion. So thank you, everybody. This was, appreciate the engagement, and I'll see people next week.